Hello everyone, and welcome to today's live broadcast, Advancing Parkinson's Disease Cell Therapy, Transplanting Cryopreserved iPSC-derived Neurons, that will be presented by Dr. Dustin Wakeman. We're excited to bring to you this educational webinar presented by LabRoots and sponsored by Cellular Dynamics International. Cellular Dynamics International is a Fujifilm company Fujifilm company and is the world's largest producer of human iPS cells and human iPS cell-derived cell types. Their growing product offering includes the iCell products, which are cells that are differentiated from healthy donors, such as the iCell neurons, iCell cardiomyocytes, etc., and the MyCell products that provide reprogramming, genetic engineering, and differentiation services from your choice of patient samples. These cells are currently being used in drug discovery, toxicity testing, stem cell banking, and cell therapy development. To learn more about Cellular Dynamics, please visit www.cellulardynamics.com. I am Susan DeLaura, and I'm from Cellular Dynamics, and I'll be the moderator for the event today. Before we begin, I'd just like to remind everyone that this is an interactive event, and so therefore I encourage you to participate by submitting as many questions as you want at any time throughout the presentation. Just click on the green Q&A button that's located in the lower corner of the presentation window and type your question in the box that appears on the screen. We'll answer as many questions as we can at the end of the presentation. Also, the presentation will be viewed in the slide window, and you can enlarge that window just by clicking on the screen icon that is located on the lower right corner. Finally, if you have any problems seeing or hearing the presentation, please click on the support button at the top right of the presentation window or use the Q&A button to let us know that you are having a problem. At this time, I'd like to introduce Dr. Dustin Wakeman. Dr. Wakeman is an assistant professor at Rush University Medical Center, where his research goals are directed at determining the long-term value of stem cell-based therapeutics in neurodevelopmental and neurodegenerative diseases. Dr. Wakeman's research is primarily focused on preclinical testing of dopamine neurons for dopamine replacement therapy, using a rational course of animal models to predict translational clinical outcomes. His lab is also utilizing pluripotent stem cells to develop new strategies for modeling disorders of the central nervous system. Their goal is to use patient-derived iPSCs as an in vitro platform to model disease-specific phenotypes and develop new druggable targets, as well as using in vivo studies to mimic human disease in the rodent and non-human primate brains. Dr. Wakeman received his BS in biology from the University of Illinois at Urbana-Champaign, and then completed his PhD in biomedical sciences at the University of California at San Diego. Please join me in welcoming Dr. Wakeman. I'll now turn the presentation over to him.
Well, thank you, Sue, for that very kind introduction, and thank you to Cellular Dynamics International and LabRoots for sponsoring this talk. And as Sue mentioned, I'm going to be talking to you today about our work utilizing pluripotent stem cells as a potential therapy for Parkinson's disease. So a quick disclosure, I am a consultant and recipient of research funding from CDI, which is work I will show you today. So I have three main learning objectives today. First is to distinguish between the different types of cell-based therapeutics for Parkinson's disease. Second will be to actually define what an authentic midbrain lineage dopamine neuron actually is. And then third, we'll be able to identify key components for a successful pluripotent stem cell-based clinical trial for Parkinson's disease. But first, what is Parkinson's disease? Well, Parkinson's disease is anatomically characterized by a specific degeneration of dopaminergic neurons within the substantia nigra, that is deep in the midbrain of your brain. And you can see in the bottom right here, in a Parkinson's disease patient, you literally have a loss of these black substance neurons. And these neurons actually project fibers or axons into the striatum, both the caudate and putamen, which is where the dopaminergic transmission actually takes place. So when we have degeneration of these neurons, we get the characteristic clinical symptoms of Parkinson's disease, things like tremor, rigidity, postural instability that you've all heard of. And again, this is a stem cell talk, so I just want to sort of characterize the different types of cells that I'm going to be talking about today. Now, at sort of the hierarchy of the stem cell, we have the embryonic stem cells and iPS cells, induced pluripotent stem cells. So these cells are pluripotent, and what that means is that they can give rise to any cell type in the body. So we're talking about heart cells, skin cells, and in our case today we'll be talking about neurons, specifically dopamine neurons. Now, if we take those cells and we sort of coax them in the neural lineage, we get what we call the neural stem cell. That cell is multipotent. And what we mean by that is it can, and at least in the case of the CNS, the central nervous system, they can make astrocytes, oligodendrocytes, and neurons. Now, these cell types have a unique characteristic in that they can self-renew. So we can make a large number of these cells from a small population. Now, if we take those cells and we further differentiate them, we get what's called a lineage-restricted neuroblast, and then we can actually move that cell towards a dopaminergic neuron, or a fully differentiated post-mitotic neuron. So these cell types are, as I mentioned, post-mitotic. So the number you start with is the number you have at the end. So really the beauty of stem cells, at least from a regenerative medicine standpoint, is that they have the capacity to both self-renew, so we can make a whole bunch of them from a small population. We can actually differentiate them into very specific cell types that we'd like to use for therapy. Now, this concept of using neural cells for transplantation in Parkinson's disease is by no means a new field. This has been going on for over 30 years. And the first cell type that really gained traction and even made it into clinical trials and showed some clinical efficacy in open-label trials was the cell we call the fetal ventral mesencephalon. So the fetal VM is the area of midbrain in the fetus where those dopamine neurons are born and will eventually degenerate during the Parkinson's disease degenerative process. Now, as I mentioned, there were some positive outcomes of these trials. However, the main issue here is that it takes about 1 to 8 fetuses to treat an individual patient. So it's extremely hard to source that type of tissue, and the tissue itself is somewhat heterogeneous. So remembering our sort of our hierarchy, our lineages we just talked about, the next obvious step is say, well, maybe we can use the dopaminergic neuroblast. And that also has shown some success in animal models. However, again, we're sort of plagued with this problem where we're still using 1 fetus to treat an individual patient. So that doesn't get rid of the sourcing issue or the heterogeneity. So if we take a step even further back, I'll be showing you some work today, previous work of mine and colleagues, where we used fetal human neural stem cells. In this case, they were derived from the subventricular zone. And that has the added advantage of you can take 1 fetus, create a neural stem cell line, and you can treat a large number of patients. And then, of course, we have the majority of this talk we'll be talking about our pluripotent stem cells, our ES cells and our iPS cells, where we can make both neural stem cells as well as a full-on post-mitotic midbrain dopamine neurons. And so really the advantage of these pluripotent stem cells is that they're renewable, right? So we can make as many of them as we need, and they're relatively, we can make pretty much relatively unlimited supply. And secondarily, that we can actually make very specific cell types. And again, for the purpose of today's talk, we'll be talking about midbrain dopamine neurons. So back in 2007, we published a paper when I was in Evan Snyder's group where we showed that after transplantation of these neural stem cells, these are human fetal neural stem cells, into the Parkinsonian primate brain, we could actually alleviate behavioral dysfunction. And so we were quite happy to find this. When we did the histology, what we found were we had these beautiful BRDU positive, TH positive cells. So these were donor cells that were labeled with BRDU, and they were TH positive. And what that means is that TH is a tyrosine hydroxylase. It's a rate-limiting enzyme in dopamine synthesis. So really, throughout the talk today, when I talk about TH, I'm primarily talking about dopamine neurons. So we were quite happy to find these TH positive cells. Unfortunately, when we counted them, there were only about 3 to 5% of the transplanted cells that actually differentiated. And we started to ask ourselves, is that really enough cells to accommodate this type of functional improvement we saw? And we thought, well, maybe something else is going on here. And in subsequent papers, we actually showed that that was probably the case, where about 95% of these cells remained as undifferentiated neural stem cells. And it looked like they were really acting through secondary mechanisms to restore and rescue the endogenous dopamine neurons that had been spared. So at the same time, when I was in Evan Snyder's group, human embryonic stem cells were really becoming in vogue. And we had some really good techniques to actually make neural cells, and not just neural cells, we were learning how to make dopamine cells. And so basically what I want you to understand here is that whenever we're working with stem cells, really what we're trying to do is model human development, okay? So when I'm trying to make a human dopamine neuron from an embryonic stem cell or an iPS cell, all I'm trying to do is do my best to recapitulate the normal developmental cues that are involved in the embryo and in the fetus to actually make these cells. And so you can see here we had a variety of different morphogens and other chemical factors, and we can actually derive these nestin-positive, SOX1-positive neural stem cells. You see they have these nice what we call rosette structures. And so we were able to make these neuroepithelial precursors. We can then add a variety of other differentiation and maturation factors. In this case, things like Wnt proteins, sonic hedgehog, and a variety of other factors. And we can actually differentiate these cells into primarily, here we have 95% beta-3 tubulin positive neurons. These are a neuronal marker. And then of these cells, about 17% actually became dopaminergic in their fate. And this was really on par with what other groups were showing in functional improvement in rodent models. So we thought, okay, well, what happens to these cells if we transplant them into our Parkinsonian monkey? And so we did a sort of a proof of concept experiment where I labeled these cells with GFP, and we transplanted them into both the striatum as well as into the substantia nigra. And you can see on these two left panels here we had very nice graft survival. And in the top right panel, these are again beta-3 tubulin positive. And you can see here in the bottom right panel, they had a very nice large, sort of this neuronal, a large soma, as well as very extensive fiber innervation. So this was great because we were showing that not only could these human embryonic stem cell derived cells survive, but they were also projecting pretty extensive fibers and innervating the host parenchyma. And this is only 28 days post-transplantation. Now, unfortunately, when I stained for tyrosine hydroxylase, I found 0 cells that were Th positive. So remember, 17% of these cells went in Th positive. So I really started to ask myself, what's going on here? Sort of where did we go wrong? Now, I mentioned previously, and I want to reiterate, as stem cell biologists, all we're really trying to do is understand and recapitulate what's going on during normal development. So really, the best stem cell biologists are really good developmental biologists. And so this is just a schematic here of the rodent brain very early during development. And really what I want to focus on here is this midline area where the forebrain and hindbrain meet. There's a gradient of sonic hedgehog and a variety of other factors. But if you take a cross-section here at the midbrain, and what I want you to focus on is on that right panel there is that little red area there. That's an area called the foreplate. And it turns out that's where these dopaminergic neurons are actually born. And in a series of brilliant experiments and very carefully thought out experiments, Ram Mackay, who's a fantastic developmental biologist, specifically in our field for making dopamine neurons, sort of figured out that there's this master regulator, FOXA2, this transcription factor that really does sort of regulate these early dopaminergic progenitors. And so you can see down in the bottom 3 panels at embryonic day 9.5, we have a very early expression of the sonic hedgehog and this LMX1B transcription factor, and then you see at 12.5, this is the beginning of those Th positive dopamine neurons. So again, through a series of elegant experiments, he really showed that by knocking this gene out, FOXA2, he could actually induce a Parkinsonian-like symptom. And so what I want you to sort of remember for the rest of this talk are these 2 genes, these FOXA2, LMX1B, and that these genes really do specify what we call an authentic midbrain lineage foreplate dopaminergic neuron. Okay, so now Ram Mackay had some extremely talented postdocs that have come through his lab, and one of those particular postdocs that came through his lab, and now is just a phenomenal stem cell scientist and developmental biologist, was Lorenz Stir. In 2011, he published this paper in Nature with Sonia Cricks being the first author. And this is, both myself and Dr. Jeffrey Cordova at Rush were actually quite fortunate to be able to work with Dr. Studer on this project. And what Lorenz basically showed in this paper was that this technique we were using, this rosette-based strategy, was completely wrong. And in fact, what we were doing was neuralizing cells towards an anterior neuroectoderm fate. And what Lorenz showed was, and again, the specifics here of these chemicals aren't really all that important right now, but what I want to point out is one specific molecule, and that's this green box on the bottom, the CHIR molecule you're seeing, and that's basically a Wnt activator. And so what Lorenz showed in this paper was that when you activate canonical Wnt signaling, you could actually induce floor plate neurogenesis. And this turned out to be a huge breakthrough and really a paradigm shift for the field. So I'm going to show you that data now. So let's focus now again on the top left panel here. There's a four-plate derived dopaminergic neurons, that technique I just showed you. And what we have here is LMX1A FOXA2 expression in these cells. And you can see the old technique, the rosette-based technique right below. You only get the Th positive cells, not the LMX1A and FOXA2 expression. Remember, those are those two critical transcription factors that are important in the foreplate, where the midbrain dopamine neurons are actually born. And to the right of that, we have Th and Neur1. Neur1 is another one of these pro-dopaminergic genes. And you can see both techniques give you Neur1 positive, Th positive cells. So while it looked like we were making true dopamine neurons with our old strategy, in fact, we didn't have the whole story. We didn't have all the signaling process. And that's just really quantified over in B in the top right. And not only were we making the right types of cells with this strategy, this four-plate strategy, but we weren't making the wrong types of cells. And by that I mean these serotonergic neurons and GABAergic neurons that have actually been implicated in some of the problems with fetal grafts. So we have expression of these transcription factors, and Lorenz very quickly realized he was onto something, and so they transplanted these cells into several models of Parkinson's disease, rodent models of Parkinson's disease. And I'm going to talk about one here just to show you the data. This is the 6-hydroxydopamine lesion adult rat. And you can see these graphs, these beautiful human NCAM positive, Th positive, FOXA2 positive graphs, indicating again that these are maintaining their midbrain lineage. And importantly, they then went on to show through 3 different independent tests that when they transplanted these cells compared to sham controls, that the animals actually had restoration of normal behaviors. You see in the bottom left panel K, a complete restoration from the amphetamine-induced rotations. We have a stepping test almost completely towards baseline again, and then the cylinder test, which is a 4-limb akinesia test. And both these stepping tests and cylinder tests, which are pharmacologically independent and more stringent, we have a complete reversal here in behavior significantly. So really what we're showing here is that not only did Lorenz's group find out that they can make these cells, but they were actually functioning. This was one of these functioning long-term, and this is one of the problems with some of the other techniques, is that they really didn't have this long-term effect. So this is about the time when Dr. Stir contacted Dr. Cordova and I and asked us if we would be interested in collaborating on using these cells in Parkinsonian primate models as proof of concept. And so, of course, we were very happy to collaborate, and that turned out to be a really good decision and sort of shaped our program from there on out. So this is 3 months post-transplantation into the MPTP lesion Parkinsonian monkey, and this brown staining on the left is human cytoplasm. So anywhere you see brown staining here is a grafted cell, and you can see in B, this is the putamen, this is the target area of the cells. You see the innervation into the putamen, as well as extensive fiber outgrowth into these white matter tracts in the corona radiata. Now, before we were not, in the studies I had done previously, I was not able to find Th positive cells, so I was quite happy when I stained for tyrosine hydroxylase and we found a large number of these Th positive dopaminergic neurons within the putaminal graft here. This is the same graft you're seeing in A. And then, of course, just as importantly, we had maintenance of FOXA2 expression within these cells, indicating, again, they were maintaining this midbrain lineage. Now, it wasn't every single cell that was FOXA2 positive, but the overwhelming majority of them were. And extensive efforts are underway in Dr. Studer's lab to actually purify this population even to an even greater extent. So what exactly do these cells look like morphologically? Well, what I want to point out here, I hope you can appreciate, that it's not just 1 or 2 cells that are mature or survive. We have large groups of these cells. You can see panel C. We have sort of a network of these cells, and they highly innervate the host parenchyma, as you can see in B, with these very long axons. And when you look in detail at these cells at high magnification, you can see in panels D and E on the right that we actually have this very mature morphology and what we call this sort of angular somata, which is characteristic of midbrain dopamine neurons. And again, you can see a nice fiber outgrowth from these cells. Now, I mentioned before that all we're really trying to do is make this fetal VM cell in a homogeneous manner and in large numbers, right? That was the whole issue with these cells. They're very hard to source. So how do these cells match up? This is an actual patient that was in a clinical trial that received fetal ventral mesencephalic cells, and you can see in A, this very nice network, highly innervated cells, and B, you can see that nice angular morphology within the soma of these cells. And compared to our cells, we actually see very similar morphology of the cells, and we also see this sort of networked innervation. Again, our cells are only at 3 months post-transplantation. So when we saw this data, we thought we were quite happy. We thought we were really on the right track. And so the next step, and that we're sort of finishing up right now, is a long-term functional study in vivo. This is a one-year study using non-human primate Parkinsonian animals. And we're really starting now, and Dr. Studer's group, through NISTEM funding, has really worked on sort of creating this final product, a GMP-compatible product from human embryonic stem cells. So we're really excited about this product. Now, one issue that we need to think about when advancing pluripotent stem cell therapy for Parkinson's disease is immune system regulation. So the idea here is that when you have an allograft, you're going to either need immunosuppression, at least short-term to some extent, or you have to do something called HLA matching, where you actually match the MHC alleles, very similar to something what you would see in a whole organ transplant. And I'll talk about that a little bit more in a minute. But that HLA matching would be much more difficult using human embryonic stem cells. So this is really when I became sort of interested and almost, I guess, fixated on using induced pluripotent stem cells. So as you know, iPS cells are derived from individuals, and of course we can then create different types of cells. I won the Nobel Prize in 2012, relatively recently considering their inception. And so now I want to focus the rest of my talk on the work I've been doing with Cellular Dynamics International. Now CDI is a rather innovative company because really what they do is they're really good at making not only iPS cells from disease patients as well as healthy patients, but they're also really good at making derivative cells, so heart cells, lung cells, and in this case, brain cells, and for us, post-mitotic neurons. And not only are they good at making the cells, they're really good at cryopreserving the cells. And so that's important because when we think about going to the FDA for a clinical trial for something like transplantation and regenerative medicine, the FDA requires you to have a very high level of QA/QC on your cells, and so it's important that you can make these cells with high efficiency in very large batches and be able to do all the QA/QC and still have enough cells left over and retest reliability to actually create a therapeutic for a large set of patients. So you've all seen some sort of version of this slide. This is my simplified version of the slide. And basically what we have is we have patient A in blue at the top. And CDI basically collects a blood sample. They use episomal reprogramming, or we call this footprint-free reprogramming, to generate induced pluripotent stem cells. And then, of course, we can take induced pluripotent stem cells and we can differentiate them into the cell type of our choice. And for us today, we'll be talking about midbrain dopamine neurons. So this is actually the product they call iCell-Dopa. You can buy this off the shelf from them right now, any day of the week. So I'm going to tell you about some of our work using these cells in sort of the right arm of this strategy, for a cellular therapeutic. I'm not going to talk today about using these cells as a research tool. However, I will say that they are very nice for use for in vitro diagnostics as well. So there's really two pathways we can look at when we think about cellular therapies for regenerative medicine using iPS cells. One is sort of the ideal situation, the autologous transplant. That is taking the cell from that patient, driving the iPS cell line, making our differentiated cell type of choice, and transplanting that back into the patient. Of course, the advantage here is, at least theoretically, you should not need immunosuppression because you have a complete match of alleles in the MHC loci. Now, the other idea here is allogeneic transplant, and that's something we call the haplomatching approach, and that is basically you use the exact same procedure that you would use in process for a whole organ transplantation, being that you sort of try and match as many of those MHC alleles as possible to get as close of a match. And so thinking about this, you could take cells from healthy patients where you genotyped them previously, you know exactly what the combination of alleles are, and then you basically pull out the allele combination that matches your patient the best. Okay, and so we're interested in both of these aspects, and we're pursuing both of them at the same time. And I think you'll see by the end of the talk how they sort of go hand in hand. So I mentioned these cells, these iCell-Dopa neurons. Well, what exactly are these iCell-Dopa neurons? So this is gene expression analysis looking, this is RT-PCR, looking at these cells at different time points post-thaw, between 7 and 42 days. And that's comparing it in the red bar here being to the human substantia nigra. And I'm not going to go through all these genes, but just a few I'll point out: FOXA2, LMX1, NER1, TH. Again, these are those genes I've been talking about that are important midbrain neurogenesis genes. And what I want you to appreciate is that sort of across this gene span, that they're very similar to what we would find in the human substantial item. So really what this says is that these iPS-derived midbrain dopamine neurons really are expressing these foreplate and midbrain specific genes. So the gene expression looks good. What about protein expression? Well, what do these cells look like in vitro? So in the top left here, you see a bright field image of these cells. This is just 7 days post-transplantation. They have these nice large soma, and they send these axons out all throughout the dish. And these will continue to grow, and like I said, they're a very nice in vitro research diagnostic as well. And of course, you can see here they're greater than 90% FOXA2, LMX1, TH positive. And again, they express TH. And when you just do a quick FACS analysis of these cells for FOXA2 and TH, you see that greater than 75% of these cells are actually co-positive. And that number is actually increased even more as we sort of CDI as we find this process. Now, one of the most important pieces of data I'm actually going to show you today is in the bottom right corner here. That's the thaw viability of these cells. So you might not think that that's really all that important of a piece of data. But when you think about trying to create this cell product and go into the clinic, this turns out to be a big deal. So before I had worked with these cells, I had never seen any post-mitotic dopamine neuron that you could actually freeze and thaw back with high efficiency. And so what you're seeing here is around 75% thaw back viability. In our hands, we're usually getting around 80 to 85%. It really depends on the user. But suffice it to say is you get a large number of surviving cells out of this vial of cells. And so that really becomes important later on when we think about trying to apply this to a patient population. And I'll touch on that in a few minutes. So again, we have this nice gene expression profile, and of course these nice pretty pictures, these immunohistochemical pictures. But when I think about protein expression, I'm usually sold by Western blots. And so this is just a few of the proteins that we've assayed through Western blots. And what you can see, the DA here represents the iCell dopa neurons. GABA here is a highly GABAergic population. This is actually another iPS line, that iPS-derived neuronal line that CDI sells, the iCell neurons, which are almost 80% GABAergic. And then we have these NSCs. These are the fetal neural stem cells. These are actually Clive Svenson's cortical neural stem cells that we've grown in dopaminergic media. And what I want you to appreciate here is that only these dopaminergic population are FOXA2, TH positive, and you see some of these other genes I've talked about, VMAT2, GERT2 being other midbrain markers, as well as having synaptic proteins. So all in all, we seem to have the right gene expression profile, as well as proteins being expressed in these cells. So that being said, the next question really is, are these cells actually making dopamine? Okay, and so this is an ELISA that was performed on these iCell dopa neurons, and as a control, these are iCell neurons. That's that GABAergic population I was just talking about. And you can see in blue, this is just the amount of dopamine that's being secreted into the basal media, in this case HBSS. And then we can also apply potassium chloride stimulation and actually get an increase in secretion of dopamine into the media. Again, suggesting that these cells are indeed making dopamine and secreting it. And we're actually in the process of validating this with HPLC and looking at the metabolites of dopamine, both homovanillic acid and DOPAC. So when I say functionality, of course, most people are going to think electrical activity, and that, of course, means doing electrophysiology. So we've patch clamped onto these cells, and you can see in A we have these cells that are firing spontaneous action potentials, and then in B, when we apply current, they actually also will fire action potentials and fire evoked currents. So it looks like these cells are functionally active, and we've taken that a step further, and we've actually shown that not only are they functionally active, but they actually have very characteristic inhibitor responses to both TTX for sodium channels as well as TEA for potassium channels. So it really looks like we have functional ion channels. And there's been quite a bit more electrophysiology and MEA data that's been generated by Kyle Mengen at CDI, as well as in Jim Sturmeier's group by Zhangzi, that we are continuing to pursue, really showing indeed that they have the right receptors and that they're actually responding in a very similar way to a midbrain dopamine neuron. So I don't have time to show you all that data today. Suffice it to say that at this point, we're quite convinced that we had the real deal, the right type of dopaminergic neuron, had very high viability post-thaw. And so the next step, of course, and what our group specializes in is testing these cells in animal models of movement disorders, in this case the 6-hydroxydopamine lesion rat and the MPTP lesion primate. And now all this work I'm going to show you for the rest of this talk has really been spearheaded by these two gentlemen, a graduate student David Marmion and a very talented technician Benjamin Hiller in our lab, who have really taken on this project full bore. And of course, our overall goal here is to figure out whether or not these cells are safe and efficacious to go for an IND approval from the FDA. So now I mentioned the ideal situation is for a neurosurgeon to be able to call down to a technician in the lab and say, I need X cc's of cells in, let's say, 30 minutes. And the technician can very easily take that vial of cells, thaw it. In this case, we actually centrifuge and wash those cells twice to remove the DMSO out of the cryopreservative, and then we are transplanting those cells directly at that point. This whole process takes roughly 26 to 27 minutes from the time we take that vial out of the liquid nitrogen to the time it is actually being injected into the brain. And so why is this so important? Because these cells do not require any subculturing, this data I'm going to show you. And that's important because we want to really minimize the amount of human exposure and human error that can be introduced into this scenario, because that's really what keeps our quality, our QA/QC, and makes this much more therapeutically viable to treat a large number of patients over a large number of different centers. So it doesn't require any special technical work or any special equipment. So that being said, what we decided to do was a very simple proof of concept. At this point, I was very skeptical that this would work. I'd never seen anything like this before. And so we basically transplanted these cells into cyclosporin immunosuppressed Sprague-Dawley rats. And this is just 2 weeks later. These are intact, healthy rats. And what I think you can appreciate here in A and B are these human NCAM and human cytoplasm grafts that are not only surviving, but they're already innervating the host parenchyma at 2 weeks post-transplantation. You can see they're also dopaminergic, TH positive, and importantly, they're also FOXA2 positive, as you can see here by colocalization with human nuclei around these TH positive cells. So we were quite happy to find that these cells not only survived and engrafted, but they also maintained their midbrain lineage. So of course the next step was to see whether or not they were safe. And the FDA, of course, is primarily and most importantly a safety organization. They are very interested in making sure that you are not going to make patients worse, and thankfully so. So when we think about stem cells, the first thing we think about for safety in regenerative medicine is, are cells proliferating, right? Because the last thing I want to do is make a tumor, a brain tumor inside of a patient, especially a Parkinson's patient that's probably going to live for 10 or 15 years at least, in which case even a few proliferating cells could be a problem. And so on the left here, you just see this is the graft zone here in human nuclei. And then in B, we have our Ki67 staining, which stains proliferative cells. And as an internal control, you see in the bottom left panel, B double prime, that's just the subventricular zone and ventricular area, which is where the endogenous neural stem cells are in the rodent brain as a positive control. And I've looked at over 50 animals at 2 weeks post-transplantation, and I can tell you I have not found a single Ki67 positive cell. It was rather hard to believe. In fact, I looked so hard, and I thought I finally found one here. You see in B prime in that inset panel where the red arrow is. I thought I finally found one. I said, aha, there is one. And then, of course, when I went and looked at high magnification, turns out that was just a piece of dust. It was an artifact from the slide. So not only are these cells surviving, maintaining their midbrain lineage, but it also looks like they have an extremely safe profile in that they're not proliferating. So we were quite happy with this. And so the next step, of course, was to go into a rodent model of Parkinson's disease. And for us, that's the 6-hydroxydopamine lesion rat. And so the model is quite simple. You inject the neurotoxin 6-hydroxydopamine into the medial forebrain bundle. That is the bundle of axons that runs from the dopaminergic neurons in the substantia nigra into the striatum where they release dopamine. And when we lesion these animals, what we find is it's a unilateral lesion. So the right side here is the lesion, the left side is intact. And so you can see in the bottom right panel B that all of these substantia nigra midbrain dopamine neurons are completely wiped out. And when we have degeneration of these neurons, we also get subsequent degeneration of their terminals within the striatum. So you can see the lack of TH staining in the striatum as well. And then from there, it's quite simple. We actually transplant the cells into the lesion hemisphere, and we see whether or not we can actually restore this dopaminergic deficit. Now, the way we know if we have a stable lesion is we can actually use this test called the amphetamine-induced rotation test. And so basically what we have is rotational asymmetry in these animals, and they'll actually, once given a dose of D-amphetamine, they will rotate, and if you get reinnervation of the cells, the animals will reduce their rotations and normalize and sort of have a zero output. And I'll show you more of that data later on. But really the important part here is that we're actually able to show that we have a stable lesion, and we're very careful in our lab. We do several retests over a 6 to 8 week period, and we have a very stringent criteria for inclusion into our functional studies. So once we had our animals lesioned and we felt like we had the right cell type, we transplanted these cells again. These are CSA immunosuppressed 6-hydroxydopamine lesion spread dolly rats. We let these cells go for 6 months, and what you can see in C and D is we had very nice survival of both human NCAM and human nuclei positive grafted cells. And I hope you can see on the right hemisphere where we've actually injected the cells, this brown staining all throughout that graft that's innervating the entire striatum, indicating that indeed we have human fibers throughout this, the injected striatum. You don't see that on the contralateral non-lesion, non-grafted side. And you can see in human nuclei that not only are these cells from our human NCAM surviving and innervating the brain, but the cell bodies themselves are staying exactly where we transplanted them in this graft zone. And that's important because when we do biodistribution studies and we're looking at safety, we really need to know where our cells are at. And it always helps when they're not moving around going funny places. So we're actually pretty happy to find that these cells really stayed exactly where they were. And you can really appreciate that they're using the human nuclei standing there in panel B. So the next question really was, well, what about dopaminergic cells? Are they dopaminergic? And so what we have here on the bottom right, again, we have our grafted cells. This is tyrosine hydroxylase staining, and you can really see now, compared to what I showed you earlier, where we had really no dopaminergic fibers, we have complete reinnervation of the injected hemisphere. And it's really important to point this out. The bottom left here is that 6 months, is that same animal. The substantia nigra is still completely lesioned. So it's not like we were rescuing endogenous cells in this process. And it also confirms that we had a complete stable lesion. So I want you really to appreciate what I'm talking about when I'm talking about innervation. So this is that same animal I was just showing you, and that was that control animal earlier that I was showing you. This is 6 months post-transplantation. And all I've done is I've taken these TH images and I've inverted them so you can really see, really brings out that stain. And so I hope you can appreciate in the top left panel labeled G that we really do have complete reinnervation of that striatum, all that blue and light stain that you see all throughout the striatum. And that's compared now to the right side where we have the 6-hydroxydopamine lesion control, and we don't see any of that innervation. So this was fantastic. We were really excited about this. But again, our job here really is not to have pretty grafts to innervate. Our job is to see if we can actually rescue functional deficits in Parkinsonian animals. And so I mentioned this amphetamine-induced rotational test earlier. And so what I want to point out here is this blue line. This blue line are animals that were grafted with these cells that when we actually did the histology on these cells, we found grafts. Some of them were quite large, some of them were smaller, but every single animal had complete restoration of normal behavior, normal rotations, the deficits that we see. And you can see in black is the vehicle control. These are animals that were given immediate injection instead of the cells. And then the red line here, these are actually animals that received a cell transplant and actually when we did histology did not have any surviving graft for whatever reason. And so this is important because you might guess that if your graft did not survive, you should not get better. And that's exactly what we saw. So the blue line actually represents an N of 6 rats. The red line is an N of 4 rats, and that black line is an N of 4 rats as well. So we were really happy to see that our histology matched our behavior, right? So if the animal had a graft, and again, it could be a small graft or a large graft, it actually got better. So this is just one test, one functional test we can do. And there's another test called the apomorphine-induced rotation. And while amphetamine-induced rotation really is a surrogate for dopamine release, apomorphine rotations really more of a surrogate for innervation, and it has to do with D1, D2 receptors. Suffice it to say, we see the exact same significant result in the animals that had a successful graft versus the lesion or non-successful graft, where we have this reversal of deficits. And we think that this trend here, you can see it's trending towards baseline. We think this would continue, and we're looking into this in our long-term studies now. So again, these were really fantastic results showing that we could reverse motor deficits using these cryopreserved neurons. And I just want to stress again that these cells were just thawed, no subculturing, and were transplanted with 2 very simple washes right back into these animals' brains. And this is 6 months post-transplantation. So, of course, we're interested in safety, as I mentioned before. So again, I looked at these graphs, and we found 0 Ki67 positive cells in these graphs. Again, you see the subventricular zone as the control. And then, of course, we wanted to measure the total number of surviving cells and the number of Th-positive cells. So in I and J, you can see really the morphology of these cells. They're extremely mature, highly arborized and innervated, sending axons out into the parenchyma. And when we counted these cells, of the total 450,000 that we actually injected into these animals, we saw around 24 to 25% on average, ranging upwards towards 30% in some animals. And of those cells, presented as surviving cells, about 35% of them were actually Th positive. And this is actually in line, in line or better than other reported groups with human embryonic stem cell derived cells, and those were non-cryopreserved. So again, these results are extremely promising, and so we were really happy, obviously, that we were able to restore function. So the next step then was really to find out what would happen in the diseased environment. So all this data I've shown you so far are using cells from a healthy individual. So this sort of would be that application, that allogeneic application, where we could do a haplotype match. But what would happen if we actually use cells from a Parkinson's disease patient, right? So there might be huge differences between deriving cells from a diseased patient and how those cells actually go back in. And in Parkinson's disease, what happens over time is these cells in the brain, the dopaminergic neurons, develop what we call alpha-synuclein inclusions. So what we're really interested right now is figuring out whether or not there are any differences between the 2 approaches, and this will really inform us how we want to go forward towards the FDA for a clinical trial. So right now, and this is work that I'm doing in collaboration with Dr. Deborah Hall in the Rush Movement Disorders Clinic. We've already started enrolling, and actually we're deriving cells from very early stage sporadic Parkinson's disease patients. So this would sort of be our target population that we would go for. And this is really a proof of concept for the amount of time it would take for us to actually go from the patient back into having a functional cell. And so it's very straightforward. Again, we're deriving using episomal reprogramming, deriving iPS cells, making the dopaminergic neurons, CDIs, then cryopreserving them, and then we're testing them in animal models of Parkinson's disease. And we've already began the derivation process and differentiation process on 2 donors, and we now have 2 more donors lined up, representing both male and female sexes as well as multiple ethnicities. So we're really interested in looking again, how do these cells compare? Are there any cross-sex or cross-ethnicity differences? To really try and get sort of a more universal idea about how our cells will function in animal models towards creating this IND package. So I just mentioned sort of our ongoing experiment with autologous cells. We are taking these cells, all of these cells, and we're going to compare them side by side in a 12-month functional in vivo study. Now, this is going to use aphyonic nude rats, so we aren't going to have to worry about immunosuppressing the animals. Animals really don't respond well to immunosuppression for 12 months. It can be somewhat toxic, and you can have other off-target side effects that we don't want to confound our experiments. Again, we're really interested in this autologous versus allogeneic approach, so looking at that haplomatch approach. How do those healthy cells compare versus our sporadic PD cells? And I mentioned it's a functional study. So when I say function, I think about things like electrophysiology. What types of action potentials are they firing? How are they firing these action potentials? Really getting in depth into looking at that type of information. And that's being done in collaboration with Dr. Jim Sturmeyer at Northwestern University here in Chicago. And then we're also very interested in really looking at the type of synapses that these cells are making, what type of cells they're synapsing on, what type of host cells they're synapsing onto them. And we're really doing that through electron microscopy as well as some array tomography in collaboration with Dr. Gan Nicholson and his graduate student Tim Musial here at Rush University. So our next step really was, after we had our functional study, was to look at safety and toxicity in vivo. And in this case, we really wanted to look at proof of concept for these cryopreserved cells in MPTP lesion non-human primates. So I just want to very briefly describe to you the MPTP model that I've explained earlier, and that you give this neurotoxin MPTP, and what we have here is a comparison between Parkinson's disease and MPTP. This is a review from Collier Labs, and as well as Jeffrey Cordova at Rush. And what we see is in both the MPTP model and Parkinson's, we have this characteristic loss of dopaminergic neurons within the substantia nigra itself, as well as the loss of striatal dopaminergic transmission, very similar to Parkinson's disease. And just as importantly, we also get the clinical manifestation, the symptoms, the behavioral symptoms like akinesia, tremor, rigidity, postural instability. So this really is what we call sort of a clinical phenocopy of at least the motor symptoms in Parkinson's disease. And so when we take these cells and we transplant them into MPTP lesion monkeys, this is 1 month post-transplantation. You can see the grafts are highly innervated. They're sending axons into the host parenchyma. You can see in A and B. Again, these are just B and C are these inverted images, so you can really pull out the fibers. And you can see in C, the cells are still somewhat bipolar, so they're kind of immature. And we've seen that previously with Dr. Studer's cells as well. And what we've also found, as I showed you, at 3 months, that those cells mature and highly innervate the brain. So what happens at 3 months? So this is tyrosine hydroxylase staining for these cells, and you can see in E and F that not only are these cells maintaining their Th-positive expression, but they also have that very nice large characteristic angular soma, as well as highly innervating the host parenchyma. And you can see in F in the inverted image on the top right panel there, that very characteristic morphology of these differentiated neurons in the high arborization there. So again, we know the cells survive, they engraft, they continue to mature, and they really are expressing the right proteins. We're actually in the process of characterizing these grafts right now. I didn't have time to put the data in, but David Marmion has just recently showed me data where indeed they are maintaining their FOXA2 expression and expressing things like VMAT2 and GRP2, which are indicative of the A9 subtype of neuron, which is particularly susceptible to degeneration of Parkinson's disease. So now, in my last slide, I just want to wrap up here. I want to just sort of show you the global view. Of course, all we're really trying to do, again, is we're trying to recapitulate normal development, and we're trying to make that cell in the human fetal ventral mesencephalon. Now, by no means has this strategy been sort of abandoned. There's actually a group right now that has a clinical trial called TransEuro, and their whole goal for this trial is not to try and bring human fetal ventral mesencephalic grafts back, but to try and learn how to actually optimize this strategy and apply, once we have all the technology figured out for pluripotent stem cells, apply those techniques that they've learned from human VM into the human situation for pluripotent stem cells. I told you about our work with Lorenz Stir's lab that's being funded by the New York Stem Cell Foundation using human embryonic stem cells. And Dr. Stir's lab is really spearheaded this project and really tackled full bore. They have now into their pre-IND process with the FDA, where they have derived their GMP cell lines that they would need to work in cell bank, and they've really done a bang-up job towards creating a cell type that could be used therapeutically. And then, of course, I've told you about our work with CDI that we're working on towards the same ends, again, using the exact same type of protocols as Studer is using iPS cells. And the idea here, again, we're trying to do an autologous cell therapy, trying to sort of get rid of some of that immunosuppression. Now, I'll leave you with this. Which cell type do I think is the best? Honestly, I don't have a clue. The point is to test them side by side and figure out which cell type has the best efficacy. Now, for any of these cell therapies to be useful clinically, and this goes for any new therapeutic, we're going to require an enhanced efficacy over the currently available treatments. And for PD, right now, that's oral L-DOPA and deep brain stimulation, which show very high efficacy for around 10 years or so. So we already have very well-established therapies for PD. So the point is not to make something that works as well as these therapies, but to make something that actually works better and creates a more long-term solution. And so I think that needs to, when we're generating these cell types and we're looking at whether or not we want to use them therapeutically, we really need to keep that in mind, that the point is not to do the same, the point is to do better. And so I'll leave you with that. I just now want to thank the people that actually done most of this work. Of course, I need to thank my mentor, Jeffrey Cordover, who's really let me go full at this project and has supported me since he brought me to Rush over 5 years ago now. And then, of course, these two in the bottom right of this picture, Benjamin Hiller there in the middle of the picture, and David Marmion, who's a very talented graduate student with Dr. Cordover, pictured on the right, that have really done the majority of this work I just showed you, and then a variety of other people from our labs, and then, of course, all of our technical help in the lab. We couldn't do it without them. And then, last but not least, all of our collaborators. I really owe a debt of gratitude to Cellular Dynamics International, particularly the two gentlemen, Chris McMahon, who is the director for the Dopamine project and really heads up operations at CDI on that end, as well as Carter Cliff, who was the first person I met from CDI that really was a champion of this project throughout and has been a huge help towards getting this to the position we're at right now. And then last but not least, Lorenz Stir's lab and Sonia Crick's really for developing this game-changing technique and really this paradigm shift towards floor plate generation of these cells. Without them, we would be still quite a ways away from where we are right now. And then all the people that helped me in graduate school, Gene Redman, Evan Snyder, my PI and mentor when I was in graduate school working with fetal cells. And then last but not least, of course, all of my funding, Rush University Medical Center, CDI, and the NIH. And with that, I would be happy to take questions from the audience.
Great, thank you, Dustin. Thank you for that exciting presentation. You know, it was really great, thought-provoking presentation. We have time for a few questions. I just want to remind everyone that if you have any questions that you'd like to ask, you can do so now. Remember to just click the green question and answer button that's in the lower left corner of the presentation window and type your question into the box that appears and press send. We'll answer as many of the questions that we can with the time that we have left. So with that, let's get started.
So thank you all for your questions. The first question comes from Maki Ogawa, and I apologize if I pronounce anyone's name incorrectly, from Applied Stem Cell Inc. And I can only see some of this question, but it says, what is the method of differentiation to dopaminergic neurons from iPSCs? Is it different from healthy and PD patient-derived cells? So the method is a slightly modified method from the Studer method. That being said, CDI does license Dr. Studer's protocol and their IP. So I can tell you it is very similar, and all of the tests that we've done using these cells, comparing them, has shown that they are extremely similar by gene expression, protein expression, etc. Pretty much every parameter we've looked at has really shown that they're almost identical. The next question was the sporadic PD cells versus the iPS cells. So I can't answer that quite yet because we're still in the process of deriving those cells. As most of you know, with any iPS cell line, there's line-to-line variability, and not only how they're derived, how they grow, the percentage of cells that actually you can differentiate from them. So they're always, no matter what cell type you're using and what source you're using, there's always going to be some fine-tuning, some tweaking, and that's one of the questions that we're actually really interested in looking at in CDI and Chris McMahon's group is really been going full bore into, is figuring out just how much tweaking that might take, coming up with a protocol or slight modifications of protocols that would decrease that variability. We do know that we can create the iPS cell lines from these cells, and we anticipate that there shouldn't be too many issues with the dopaminergic differentiation itself and the cryopreservation. I know there's other groups around the world that have done the same, they use a similar approach and have had success. So I don't anticipate any issues there. So thank you very much for that question. That was a great question. Let's see here. We have a question from Suji Sebastian. Would the cryostep be repeatable in a real world at a clinic on another continent? So I think you're talking about the actual cryopreservation of the cells. Is it repeatable? It's extremely repeatable. This is not one lot of cells that I'm talking about. Cellular dynamics generates these cells all the time. Lots of lot variability is next to none. They have, I can tell you, they have a very strict release criteria, which is something that's very important for scientists like us, right? Because we want to make sure we're getting the same thing every time we use the cells. So that release criteria, not only is it strict, that's a good thing for them because they have a repeatable product, but it's great for us because we know our experiments are not going to change depending on the lot we get of the cells. And I can tell you I have tested multiple lots of these cells in vivo, and I always see the exact same results. And that was actually one of those questions you have as a scientist is you know, is this going to be a one-time thing? And the answer to that is not. The next question I see incoming is Shakar Diao from Irvine Scientific. Have you compared DMSO containing versus DMSO free conditions post-thaw in your functional parameters? So the simple answer to that is no. We have not tested non-DMSO containing conditions, and the reason for that is that I'm quite happy with the results we're getting with the DMSO containing cells. There are other cell products for other diseases using other cell types on the market through other companies that are in the clinic that are cryopreserved, having DMSO in them. The real situation that you need to worry about here is whether or not there's residual DMSO in the preparation when you transplant. And that is something that we can very easily test. We can measure that. And as long as you're underneath that sort of criteria that you set that's safe, that's been set, and the FDA has guidelines on that, and there's other people that have looked at that, then the prevailing thought is as long as you've done all your safety and safety studies, you've shown that it's safe, then that's not a problem. So you know, I guess it would be interesting from an academic standpoint whether or not it works without the DMSO. We're very happy with our results with the DMSO, and we don't think it's going to be a roadblock to the clinic. So I guess for me it's one of those, if it isn't broke, don't fix it. Just be aware of it, make sure it's safe. And since there are very simple ways to remove the DMSO to a sub-threshold level that we would need for toxicity, that's pretty much been our approach. But that's another great question. Thank you. So the next question I'm seeing is from Neil Reiner at the University of British Columbia. What is your best estimate of the timeline for entering phase 1 clinical trials, and what are the major hurdles to getting there? Wow, okay, that is a fantastic question, Neil. Estimate for timeline for entering phase 1. We do a lot of this modeling and looking to try and figure out, you know, what's the minimal amount of time, what's the maximal amount of time. I wouldn't put any set amount of time on that, and I know that's not answering your question. I'll give you some basic parameters. I would say, an ideal situation, if everything works, 3 to 5 years. I think I know CDI, we're very, very concerned with safety and doing it right. Not necessarily being the first person to do it, but doing it right and being able to reach a large patient population. Remember, I mentioned that we'd like this to be available, you know, really throughout the world eventually. So you don't need a specialized center or a specialized technician. So estimate for a phase 1, I mean, it could be as quick as 1 or 2 years if everything went perfect and the FDA was satisfied with all of our preclinical studies. There's other groups, certainly. Lorenz Stir's group is doing a bang-up job right now with their ES-derived cells, really setting the bar, I think, for all of us in the PD field. There are other people in the PD field that have cell products, I think, that would be reasonably test. I don't know as much about them, of course, personally. There's a group led by Jun Takahashi in Japan that is probably closer to the clinic than some of the rest of us, and he's using iPS cells as well. Now, as far as the major hurdles getting there, I actually have a whole slide on this. I took it out last night. So I'm sort of regretting that. But that's the great question. So when you design a clinical trial, especially for regenerative medicine, there are some major roadblocks in the way. And when you look at sort of one of these timelines, it's a cyclical timeline. It's not like a line by line horizontal line to the clinic. So one thing I've learned in the last 5 to 10 years working with stem cells and thinking clinically is that you really need to design your clinical trial before you do your preclinical tests. And so careful design there will save you a lot of time and, in reality, a lot of money, so you don't have to repeat yourself. And so what do I mean by that? Well, for any of these cell therapies, and I don't care which disease you're working on, this is going to be the same, your product has to be very carefully designed. It has to be safe, it has to be repeatable. The QA/QC that the FDA wants is extensive, and rightfully so. That's for all of our advantage. So this is one thing I think that I really learned and I admire from the Studer group is their attention to detail creating master cell banks. So these have to be GMP compliant. You need a master cell bank, a working cell bank. And so when you take these cells and you bank them, that's the cell type, your final product that has all your QA/QC that you have to go back and do all your preclinical tests on. So you have to do your efficacy tests in rodent models of disease. You need to do tumorigenicity tests, probably in a large animal model. If you're going to immunosuppress in certain cases, if you need immunosuppression, you're going to need to look at the safety profile there. So, again, you need safety, and then you need safety, and then you need safety. And one always hopes that you're also efficacious as well. But the last thing any clinician wants, and any of us working on this want to do, is take a diseased patient, someone that's sick, and make them sicker, or possibly give them a tumor. That's just not acceptable. So everyone that I know that's working on this throughout the world, whether that be in Europe, in Lund, there's a very strong group led by Malin Parmar, people like Anders Bjorklund, people like Steve Dunnett, Roger Barker over in Cardiff in the UK, Tito Knuth, all these people, the Japanese group, Jun Takahashi, Lorenz Stir's group, they get together once a year actually, and they have a meeting called the G-Force, the PD G-Force, and basically everybody gets together and talks about what they're doing, and it's really a unique approach where everyone that's competing with each other is actually working with each other to some extent, so that we are learning from each other's mistakes, and we're all learning how to get better at this. So it's really a universal good. So that is sort of a long, roundabout answer to your question. I really thank you for that question. It was fantastic. And it's a complicated question. And I think at the end of the day, what you're going to see is the people that are doing it right are very systematically going through this process and learning from the FDA what they want. And I should probably point out that it's not immediately clear. The FDA doesn't have a set of guidelines where you must do, A through Z and we'll approve this. It's different for every application. So great question. I see another question here from Suji Sebastian from, I apologize if I botched this, Loughborough University. So, do you consider a risk-based approach, EMA supported, to take the product to clinic? I'm going to take the roundabout answer on this. So, really, my job, as far as I'm concerned, as a research scientist, my job is to test the hypothesis in these animal models of Parkinson's disease. That's sort of my forte. When it comes to going to the clinic, I really, I know my area of expertise, and I know what areas I'm not an expert in. And one of those areas I'm not an expert in is, actually implementing to the clinic. So we have a lot of different advisors, of course, people that were formerly on the FDA, and this is going to go for any group. So I really rely on the support of people in the pharma industry that have been through this before, other people that are working on cell-based therapeutics, and consultants that know how this process exists. And so they can inform me as to how to design my basic studies, my safety and efficacy studies with the final product, and creating that final product, and then really handing it off to them to implement that. And I think one step that everyone needs to keep in mind is that you need to work hand in hand with your clinicians. Understand who your patient population is. Who are you targeting? What are the symptoms you're targeting? You need to have a good working relationship with your neurologists and with your neurosurgeons. And that's one thing, and really the reason that I'm here at Rush is because we have such an outstanding movement disorders clinic that knows how to implement clinical trials. So, really the answer to your question is I leave those questions and sort of those approaches to the experts in that realm and that domain, and I let them inform me and talk to those experts who would actually be able to bridge that gap, whether it be in the pharmaceutical industry, biotech, regulatory, or in the clinical realm. So again, a very nice question. So I'm looking at the Q&A, and I'm not seeing anything really on the board here. So I think I'd like to thank everyone that attended this conference. It was a great honor. Thank you to Cellular Dynamics and to LabRoots for hosting. And again, we're really excited about this work here that we're doing at Rush in collaboration with CDI, and of course all of our collaborators throughout the world. We're really pushing forward to try and help the patients. And at the end of the day, I think that's the key here. That's the goal, is to understand that we're doing this for the patients. So I guess I'd like to just finally conclude with thanking those patients, especially our donors that we're getting those cells from right now, so we can hopefully try and create a better therapeutic for them. So thank you very much for your time, and I think we'll wrap it up with that.
Hi. Once again, I'd like to just take a minute to thank Dustin for his great presentation and for today, and hope that everyone enjoyed it. For anybody that had additional questions that were not answered, please feel free to contact us at cellulardynamics.com, and we would be able to help answer your questions or get you in touch with Dustin to be able to further expand on some of your questions. One last note, I wanted to let everyone know that today's webcast will be available on demand through viewing through LabRoots for 6 months from today's date. You will receive an email here shortly letting you know when this webcast will be available, and feel free to share that email with your colleagues who weren't able to join us today. Thank you for joining us, and we hope to interact with you again soon.